That's the beta-adrenergic receptor. Let's call it the beta-adrenergic receptor. It's unique in its primary sequence. That's why it's called the beta-adrenergic. The adrenergic term is because it is activated by epinephrine or adrenaline or norepinephrine or noradrenaline. Okay? So that's the adrenergic term. So when there is here's epinephrine there, activates the receptor, changes its conformation, which in turn activates the G protein. Here's the G protein that you want to kind of be familiar with. This G protein is made up of 3 subunits, designated alpha, beta, gamma. The reason we call it G protein, because it can bind the nucleotide GDP. It can also bind GTP. So when the receptor associates with this G protein, it causes 2 things to happen. The first one is the alpha subunit here dissociates from the other 2 subunits, beta and gamma. The second thing that happens is the GDP that was bound to the alpha subunit is replaced by GTP. So what you end up with is this single subunit, Gs-alpha, with GTP bound to it. You see these sort of things here. This is the active G protein. This is the only form a G protein is active in. The alpha subunit with GTP bound to it. So it's designated small s because this is a stimulatory G protein. So Gs-alpha with GTP bound to it is going now to bind the target enzyme or effector enzyme I mentioned before, adenylate cyclase. Adenylate cyclase is going to take ATP. You notice that adenylate cyclase is part of the membrane, but whatever it produces is going to be produced inside the cytoplasm. So it takes ATP and cyclizes it, hence the name cyclase, adenylate cyclase, to produce this pink structure here is cyclic AMP, the second messenger produced by the beta-adrenergic system, or the adenylate cyclase system. You with me so far? Okay, you need to know those details. Everything I just said you need to know, okay? Second thing, this is the only second messenger produced. First of all, cyclic AMP is the only second messenger produced. Cyclic AMP is going now to activate a protein kinase. And if you're not sure what a protein kinase is an enzyme that is able to phosphorylate other enzymes or other proteins on specific residues. Typically, these kinases will phosphorylate a different type of amino acid. They will phosphorylate serine residues or threonine residues. Just keep that in mind. So cyclic AMP activates a protein kinase A. The A is dependent on cyclic A. That's basically, if you want to remember the name of the protein kinase, just remember cyclic A. It activates protein kinase A. Now this has many cellular substrates. And it's going to phosphorylate many enzymes on specific residues and change the activity of these enzymes, either negatively or positively. Okay? So you need to remember the stimulus, the type of receptor, the type of G protein, the active form of the G protein, and the effector enzyme, the second messenger, and then the second messenger activating protein kinase A as the last step. There's a little more detail here. This is just the G protein by itself, and I just want you to, let's say this G protein was activated by the receptor, and here comes GTP. It kicks off the GDP, replaces it, and it binds to the alpha subunit after it has dissociated from the beta gamma. This is the active form of the G protein, Gs-alpha. Importantly, though, I want to just say to you that this Gs-alpha and all alpha subunits of G protein have an enzymatic activity. That enzymatic activity is called GTPase. GTPase meaning it can hydrolyze GTP, and when you hydrolyze GTP, you return it to GDP. This form is no longer active. The alpha subunit reassociates with the trimer, with the beta gamma, and the activation of adenylate cyclase is terminated. So what's very important to appreciate, this is a shut-off signal, a termination signal to the adenylate cyclase system. You turn off the activation of adenylate cyclase by hydrolyzing GTP back to GDP. This form can no longer activate adenylate cyclase and produce cyclic AMP. Okay? It's very important to appreciate that in all cells, when you activate something, you have to have a turn-off mechanism. It's kind of like when you turn on the light, right? I mean, if you keep the light on, the bulbs won't last as long. So same with the cell. If the cell is, usually these hormonal effects on receptors are short-term effects, you know, for minutes or seconds even. So you want to have a turn-off mechanisms. This is a turn-off mechanism. Part of the GTP, the Gs-alpha, has a GTPase activity that can hydrolyze GTP back to GDP and terminate this entire process. Okay? And here's another one, kind of more, but really we've gone through it enough. I just want you to appreciate that the effects of cyclic AMP. So before receptor activation, there was no cyclic AMP. This is what I want you to appreciate. After receptor activation, cyclic AMP goes up. So the only reason you have cyclic AMP is due to the activation of adenylate cyclase by G-alpha S or Gs-alpha. Okay? Cyclic AMP is an allosteric, if you want, activator of this enzyme, pKa, protein kinase A. This has many substrates that can phosphorylate enzyme and change their activity. Importantly, you note the color change. So this is green. That's specific for epinephrine. This is turquoise. That's specific for glucagon. A different receptor needs a different stimulus. The glucagon receptor here will be activated by glucagon only. Cannot be activated by epinephrine or norepinephrine. However, on the intracellular side, everything is identical. Gs-alpha, adenylate cyclase, cyclic AMP is produced, and pKa is activated. And again, finally, a reminder that the alpha subunit has GTPase activity to hydrolyze GTP to GDP to terminate this entire process. So I'm going to give you a quick reason here. The reason you have this redundancy, two different receptors stimulating the same pathway, because these receptors have different tissue distribution. There may be more glucagon receptors on one tissue and no beta-adrenergic receptors. Conversely, you may have a more wider distribution of beta-adrenergic receptors on many tissues, but no glucagon receptors. Okay? So that's the importance of this redundancy.